In this video, I'm going to look at sampling schemes and manipulative experiments and compare and contrast, looking at some of the advantages of manipulative experiments over simple sampling schemes. And I'm actually going to use some of my work from my PhD, which I did quite some time ago. And these four images give you some idea of what I was working with. I was actually looking at the plants and animals that live on boulders in intertidal rocky reef areas. So up on the top left is one of the two places I worked, the Cape Banks Rocky Shore, which is on the northern side of Botany Bay. To the top right, that's the Long Reef Rocky Shore, which is in the northern suburbs of Sydney, over the Harbour Bridge and some 40 minutes north of Sydney. Down to the bottom left, the green stuff there, you can see some seaweed on the rocks there, and the green stuff that you see on the side is ulva. We commonly called it sea lettuce because it looks a lot like lettuce. And then finally, bottom right, you can see some of the animals that typically lived on the underside of the boulder. This large guy here is actually a mollusk, a snail. And then much of the white material here is the shells of marine worms that live attached to the rock and filter feed. And then there's some stringy algae and sponges and other kinds of creatures. Now, one of the things I did was monitor the arrival of animals and plants on the seashore. And I did this by taking large rocks from high on the shore, where they've been scoured by the sand and the waves and baked by the sun, so there's absolutely nothing on them, and moved them down into the intertidal area. And the graph here is showing results for one of the animals on the underside. One of those tube worms, Galliolaria. Top two panels here are for sets of rocks put out at Cape Banks, and the bottom two panels are for sets put out at Long Reef. And the main difference is one set is put out in the winter period and the other in the summer period. And the different lines are just for sets of rocks put out in slightly different locations, slightly different sites. Now, one thing you can see, particularly if you compare the top panel to the third panel, is that this animal was consistently more abundant, more common at Cape Banks than at Long Reef. This is especially true if you look at the scale, because it goes up to 3 here and it only goes up to 0.9 here. The same was true in the summer period, but not much arrived anywhere during that time. So if I go over here and generalize, the Galliolaria worm on the underside was typically more abundant at Cape Banks than at Long Reef. Now I can look at the other species. Also on the underside were spirorbits, tiny little worms, and there was no particular difference. They were pretty much the same at both places, and so was this little barnacle, Tetraclytella. If we look at the tops of rocks, really only one thing arrived there, this sea lettuce, and it was also more common at Cape Banks than Long Reef. So if I'm thinking in terms of the biology model testing process that is outlined in topic 1, what I've got here is a set of observations, a set of 4 observations about 4 different species of marine organisms. 2 of these observations are not especially interesting. They just say that there's no difference between the 2 rocky shores. But the other 2, I find a clear and consistent difference, with the species being more common at Cape Banks than at Long Reef. So the second stage here is clearly to build a model. And what I've done over here in the left panel is give you a bit more information and put together the general model. So if you remember back to the original images, and let's just go back, the Cape Banks rocky shore has orangey red and sometimes pale whitish rock. It's sandstone. In contrast, it's a bit hard to see in the photo because of where the light's coming from. In contrast, at Long Reef, the rock is dark grey to black, and it is shale. These are both sedimentary rocks, but they're quite different kinds of rocks. So let's move back here. So the sandstone has large grains, the shale has very fine grains. And as I've written, there might be differences between the physical properties of these rocks that affect the survival and growth of the plants and animals. And also the difference between the top and the bottom rock may mean that we get different things, see different things for different species. In the right panel, I've indicated what the model is for each species. So for ulva and for Galliolaria, sandstone is better than shale, and presumably, as I said, this is due to some of those physical properties. For instance, black will heat up faster than lighter colors, and so the black might be less suitable for the alga. Sandstone being grainier might be easier for the worm to attach to. Now for the other species, I should not expecting anything much to go on because they don't seem to care. They don't seem to mind. Next stage is the hypothesis. So this is where I think about a prediction. And I'm going to do a manipulative experiment. I'm going to change things. Now earlier, I mentioned that there are stores of bare rocks up high on the shore in both places. So I'm going to go to Cape Banks and I'm going to grab some of those and move them down as I've done previously. And I'm going to take some over to Long Reef. And at Long Reef, whoa, I'm shaking a bit here. At Long Reef, I'm going to do the same things. Take some down to lower on the shore, intermingle them with the transplanted sandstone rocks, and take some over to Cape Banks and intermingle those with the sandstone rocks I've brought down from higher on the shore. What this means is I will have experimentally created a situation where I can observe colonization on both types of rock in both places. This is something that doesn't occur naturally. Over on the left, you can see my predictions. Now, because of limited space on PowerPoint, I've abbreviated these, but I should see better survival and growth of the seaweed, the sea lettuce on the sandstone and then on the shale in both places. And I should see better survival and growth or establishment of the worm on the underside in both places. And yes, I did actually do this. I loaded rocks up into the back of the station wagon and drove them from place to place. And I'm talking fairly large here, so up to about 30 or 40 centimeters in diameter. Okay, so what happened? Here we go. This is the actual diagrams from the paper. Now, I'll talk about these second set of results here for Cape Banks for the animals a little bit later on, but at this point in time, don't pay too much attention to them. So what you have in the top row is results for about 3 months, and then results for adding in about another month and a half or so for the 4 species. So let's go over here and look at the sea lettuce after 3 months. Wow. The white bar is for the sandstone, the black bar is for the shale, and you can see clearly at both places much better survival or growth or establishment on the sandstone than on the shale. And at Cape Banks, the difference is extreme, nearly 40% on the sandstone and hardly any on the shale. A similar difference over at Long Reef, but here it's getting close to 90% on the sandstone and about 45% on the shale. Now, as it happens, Long Reef is the first place I want to check, and when I walked down there, the difference was visually striking. Nearly all of the top of the sandstone boulders was covered by sea lettuce, but only about half of those shale boulders. Now, if you go down a month and a half later, the stuff has started to die off a little bit, or it's getting eaten, but the pattern holds. So, if we go down to conclusions there, abundance greater on sandstone, the predicted effect observed model gets a big tick. Okay, skipping to tetraclitella, look at the numbers there, 0.06 per centimetre squared. Basically nothing arrived. The error bars here are large, so there was no statistically significant effect. So, abundance equal on both, no effect observed as predicted. Now, if you're reading the conclusions down the bottom, you will be seeing a little bit, I think, where I'm going with it. So, what happened to Galliolaria? Well, at 3 months, nothing arrived at Long Reef, but some did turn up at Cape Banks. Now, 0.2 per square centimetre doesn't sound like much, but these rocks here are 30 to 40 centimetres in diameter, so there's a reasonable number of these animals arriving on the rocks. But, contrary to expectations and predictions, there's statistically significantly more on the shale. So if you go to the third line, you'll see there was an effect, but it was opposite to the effect I predicted. Oh, so that particular prediction and that particular model gets a big cross. Something was going on, but it was way wrong compared to what I was thinking was going on. Go over to the spirorbits. Here I actually said nothing is going on, and oops, wrong again. Nearly 3 times as many of these on the shale at Cape Banks than on the sandstone. And I said nothing would happen. Also, a bit of a difference at Long Reef in the same direction, but the error bars are quite large, so that's not statistically significant. Down here at Long Reef, it is. And again, there's more on shale than on sandstone. Okay, so out of my predictions here, I'm doing, let's say, average. I got 2 right and 2 wrong. And in one case, I predicted an effect, which did happen. In another case, I predicted an effect, but it turned out to be the opposite of what I predicted. In the other 2 cases, in one I predicted no effect for the barnacle, and that's what I observed. For the spirorbits, I predicted no effect, but there was an effect, and it was the same as for Galliolaria. Okay, let me come back here to Cape Banks in the second sampling time. Why did I say ignore those results, or I would comment on those results? I had to mark my rocks with little labels so that I could identify the ones that were in the experiment. And when I came to Cape Banks on that particular occasion, I found that all the rocks had been moved and gathered up together in a single location. Now, it's actually highly likely that that disturbed the underside of the rock, so I don't regard the results on the underside as reliable for Cape Banks, because the animals were probably squashed. The alga on the top, however, being soft and floppy, and also being on the top and not on the bottom, appeared to be okay, and the results where I got at Cape Banks there were consistent with the other 3 results. So that looks alright. Now, that does, however, mean that my results for Galliolaria is resting really only on one set of results there, so I'll call that tentative and say I'd want a bit more confirmation. For tetraclitella, nothing arrived, so I can't really say anything much about what's going on there. For the spirobids, I've got a result at Cape Banks at 3 months and a result at Longreef about another 6 weeks later. So I'm reasonably confident of that. And adding to my confidence is the fact that a couple of years later, an honours student took up this particular observation as their project, and they confirmed that result. So what they were trying to do was find out why that was happening. So they tried different kinds of textures and different colors, and they couldn't actually work out what the actual physical process was, but they confirmed that it was happening. Okay, to sum up, the observations I initially made gave me some idea what was going on, but that information, those suggestions, when tested, weren't always reliable. And so that's why you'll commonly hear people say causation is not correlation, or correlation is not causation. A pattern may indicate what's going on, but something more complicated might be happening, and that's clearly what was happening here. Second important point, the experiment unambiguously worked out what was going on for 3 of these species. Because of disturbance to the experiment, the results are tentative for the Galeolaria worm, and because hardly any of the barnacle arrived, we can't really draw any firm conclusions there at all. But for the sea lettuce and for the spirobids, there are fairly strong and convincing results. Now, what might be going on here that's more complicated? Well, let's go right back to the start, to the photos. And if you look up here, you'll see that, first of all, there's a lot of seaweed growing around here at Longreef, which there isn't at Cape Banks. And then you may also get glimpses of sand. So the area at Longreef was very sandy. So the bottom of the rocks here are often resting on or in sand, or fine sand and mud. And that's clearly a different environment to what's going on over there at Cape Banks, where there's not much sand there at all. So the processes happening at these 2 places are different. And that's why the experiment here is powerful. It allows me to put both types of rock in both locations, so I cancel out the difference between the 2 places in the other environmental factors. The last thing I should say about this experiment, and this is 100% true, is that I thought it was a waste of time. Yes, you heard that correctly. I did it because a colleague who was doing his PhD at the same time was interested in the effects of a rock type, and he asked a question about it in my first seminar on my project. I got about halfway through my project and suddenly realized, when I was going through my notes, that I hadn't done anything about that particular question. And I knew he was going to be at my next seminar, and I knew that if I didn't do something about it, he'd be asking questions. So I basically did this experiment to shut him up. And when I walked down to Longreef on that first sampling occasion and saw that, I was staggered, because I thought nothing was going to happen. From then on, I've been very cautious about drawing conclusions. Kind of easy to be wrong. Okay, that's another example of going through the observation model hypothesis and results process. And hopefully it gives you some idea of the strengths and weaknesses of sampling versus experiments. If I make a final point, sampling programs are generally easy to do, and you can do lots of places. As you saw, I did 2 different times a year. Go back. 2 different times of year, 2 places, 3 sites at each place. Experiments can usually only be done on a smaller scale. So there was literally 2 lots of 8, 16 rocks involved in this. 32, because there was 8 of each type in each location. So it's either 16 or 32. It's so long ago I can't remember. But it's a much smaller scale than in the sampling study. So we've got a balance of scale versus powerful testing of hypotheses. The end.